Bismillahirrahmanirrahim. Welcome back. This is Muhammad Tariq and you are watching my YouTube channel MCQs Journey. And today video lecture we will discuss about technique used in cell biology class 11th and at the last we will discuss some MCQs related to that topic. So watch the video till the end. Let's start. Technique used in cell biology, it means that the cell is a smallest particle of our life. The cell is a building block of our life. So if we want to see the cell, if we want to see organelle inside the cell, we must need to use some technique for that. Microscope is an instrument we are able to see, but some cells are transparent, some organelle are transparent, and we cannot be able to see those transparent things on under the microscope because the light directly pass and we not able to see under the microscope these things. So we use some technique to see those objects under the microscope. And which number one is staining. Staining is a process which we will discuss later. And number two is centrifugation that we will discuss in the next lecture. Centrifugation means making parts. We isolate the different part of the cell from each other. Number two, tissue culture is also a process through which we sell our tissue. We grow outside from the body of organisms. And chromatography is also a process, electrophoresis and spectrophotometry and microdissection through which we cut some part from the cell and we see under the microscope. So this is all about technique used in cell biology, but in today's lecture we will only discuss about staining and some MCQs related to staining process. Staining means colorize of something. To colorize something is called staining. Staining, a technique that is used through which we colorize a specimen to see under the microscope. This is known as staining. It is a process through which we see a specimen under the microscope and colorize a specimen. Specimen means anything that are under the microscope and we see anything that we see under the microscope is known as specimen. Okay, maybe cell, maybe in hospital, blood, maybe different type of thing that we see under the microscope is known as specimen. Importance of specimen, I first tell that the technique mean that to make the transparent object to visible under the microscope. So staining importance mean to visualize a thing, to see the thing under the microscope, maybe organelle, maybe cell, maybe a different part of our body, everything where some distraction occurred, we see under the microscope. There are different type of staining on the basis of, there are two types on the basis of layer. Number one is single stain. Single, a type of stain in which we use only one color to visualize, to see a thing under the microscope is known as single stain. Okay. Number two, double stain. A type of stain in which we use two color and which we see and which we use two dye to mean two colors to see a thing under the microscope. There are two types. The first color is known as principal stain. Principal stain. And number two is a counter stain. The second color is known as counter stain. Number one is a principal stain and number two is a counter stain. Okay. Another type is vital stain. Non-vital. A type of stain that we use for living thing, or in simple word we can say this, the type of stain which when we use on a specimen, so the specimen will not die. This is known as vital stain. And mostly the vital stain we use for microorganism. For example, If I take one example is sapranine example. Sapranine is a type of dye that we use for bacteria to see bacteria under the microscope, and we visualize and we understand that what type of bacteria is that. Is that bacteria is a gram-negative bacteria, or that bacteria is a gram-positive bacteria? So we use sapranine, and we will discuss later what is gram-positive and gram-negative bacteria. Number 2 is a vital non-vital stain, a type of stain which is used for non-living thing. Mostly we use in hospitals. For example, if a blood is black due to leaflet, due to different type of things. When a blood vessel is blacked, so we use non-vital stain, and doctor applied these non-vital stain on in our blood vessel. When doctor applied this and this blood vessel, doctor take and understand that where is blockage occur. So we use non-vital stain for what? We use non-vital stain for hospitals and laboratories in different places where we do experiments, where we do tests, different places. Okay. And another types that is permanent stain and temporary stain. Permanent stain is a type of stain when we once applied, that stain will not finish, that stain will not be able, we not able to remove. And a type of stain that we not able to remove is known as permanent stain. Number 2, temporary. Temporary stain, a type of stain that can be removed after once we applied. So now come to MCQs that is related with staining process. Okay, so come to MCQs. MCQs number 1, vital stain is also called what? There's a very much important MCQs for MDK test that you must need to remember. The vital stain is also called in vivo stain. The second name of vital stain is also known as in vivo stain. And now also this like in vivo stain is also called what? Vital stain, opposite. And non-vital stain is also called in vitro stain. In vitro stain mean that we use for non-living thing. Why this is called in vivo stain? Because that we use for living thing. Why this is called in vitro stain? Because that we use for non-living thing. MCQs number 3, which bacteria is appear purple violet color after staining? So gram-positive bacteria has gave us purple violet color after the staining, because it is more peptidoglycan as compared to gram-negative bacteria. So thanks for watching, and the next video we will discuss about tissue culture process, centrifugation process, inshallah, and see you in next video. Goodbye. Subscribe my channel please. Okay, bye.